All right, hello everyone. So this is the video for the spinal nerve and nervous system chapter. We'll try to go through this relatively quickly. I know a lot of you might have some prior experience with this material, but if you haven't, that's perfectly fine too. I'll treat this as if this is the first time you're hearing this material. And I am Ryan Dickerson. I am a Brody student in the class of 2025, and this presentation is made by the Student Collaborative Resources for Understanding and Brody Success, so the Scrubs team. And if this is something that you are interested in getting involved in, we 100% would love you to be involved at the end of the semester. So, quick mission statement for Scrubs. Scrubs is a student-driven initiative that aims to develop resources for current and future cohorts that will pass through Brody. Members of Scrubs, we participate on a variety of subcommittees to work and create resources for students and inspire the students. And the aim is to give you a perspective from us about resources that we wish we had been exposed to during our time in the course. So if this is something you're interested in, again, we invite you to join us as you reach the end of your semester. So with that being said, disclaimer, the resources that are included in this document are made by students and not faculty, so there is the possibility for errors in development, although we do have a team approach that goes through multiple stages of vetting, so that will hopefully be diminished as much as possible. If there is a contradiction in the coursework, please go by your course documents. And again, this is a supplemental resource, so it's not replacing the instruction by the Brody faculty, and use this as a supplement as opposed to a primary resource, as we do not include all of the material that is presented in your course. So, getting to the actual material, we're going to start with the basic anatomy of the neuron. So the neuron is a nerve cell, and it is the unit of structure in the nervous system. So neurons can be various shapes and sizes, but in general, there are a couple of main components. You have a singular cell body, and the cell body is the location in which you will find the nucleus of the neuron. You will have an axon, so a singular axon. This is where you're going to be sending a nerve signal from the neuron to another structure, possibly another nerve, and you can have one or multiple dendrites. So the dendrites are a receiving signal from another neuron. That signal is getting integrated within the cell body. And then if it is deemed that a nerve signal needs to be fired from this neuron, it will be sent down the axon towards the axon terminals, which are the ends of the neuron. Now, I want to point out that in the peripheral nervous system, myelinated neurons are myelinated by Schwann cells. So myelin is a way that we insulate nerves. It allows for the nerve conduction speed to be increased, meaning that a signal is sent a little bit faster. In the peripheral nervous system, that is done by Schwann cells. Right. Now, I'll touch on this a little bit more in just a moment, but you can see from this image that we have a effector neuron, which is a multipolar motor neuron, and a sensory neuron, so a pseudo-unipolar sensory neuron. I'll touch a little bit more on how these work in just a moment. But some confusing points that your course pack makes that I just want to clarify, that even though the nucleus is within the cell body, a group of cell bodies in the central nervous system is called a nucleus. So a group of cell bodies. So a group of these cell bodies within the central nervous system is termed a nucleus. And then a group of cell bodies within the peripheral nervous system is termed a ganglion. And again, we'll mention this again a little bit later on in this video, but knowing the difference between the nucleus of a cell body and a collection of cell bodies within the central nervous system being a nucleus, or in the peripheral nervous system being a ganglion. Some other quick definitions: a nerve fiber is the axon plus its coverings. So that's going to be the axon plus the myelin sheath. A nerve is a collection of nerve fibers bundled together. So moving on to signal transduction. So this is actually how we send signals using our nerves. So again, we talked about signaling coming in to the nucleus. We're integrating the signal to determine if we need to fire the nerve, and once it does, it is going to fire in only one direction, down this axon. So it travels down this axon towards the axon terminals. At the axon terminals, this is amplified or zoomed in over here in this image. We see that this is the nerve that was sending the signal. So this is going to be the presynaptic nerve. So the synapse is where we have the two nerves coming together. So presynaptic and postsynaptic. nerve fibers. So presynaptic nerve fibers send signal down, and we're going to get to the release of neurotransmitters into the synaptic cleft. So the synaptic cleft is the small, really, really tight space between the presynaptic nerve and the postsynaptic nerve. And this is going to allow for us to have signal transduction from one nerve to another. Now, going over here on the right, I want to take a moment and talk about the difference between the multipolar motor neurons and the pseudo-unipolar sensory neurons. So the multipolar neurons are made up of a cell body that has multiple dendrites that are receiving signals from various locations with a single axon sending an output. And these are the nerve cells that will be seen in motor nerves, or nerves that are effector in essence, meaning that they are sending signal from the central nervous system. Now, the pseudo-unipolar nerves are typically your sensory neurons, and these have a singular process, so it's kind of hard to say dendrite versus axon here, but you're going to be receiving signal from some organ. This could be the skin, your tongue, your eyeball, but you're getting a signal, sensation coming in, and it goes down the axon to the central nervous system. Now, the reason these are called pseudo-unipolar is because it looks like it's unipolar. There's one fiber going through, but the cell body is hanging off the sides of this appendage. So this is in the peripheral nervous system is where we're going to find a collection of cell bodies. So a collection of cell bodies in the peripheral nervous system is going to be called a ganglion. So you're going to see, we're going to talk about a little bit later, the dorsal root ganglion that is going to refer to a collection of these specific cell bodies of pseudo-unipolar sensory neurons in the peripheral nervous system. So collection of cell bodies in the peripheral nervous system, once again, is termed a ganglion. Now, going into a little bit of the organization of the nervous system, I first want to talk about the CNS versus PNS. So that's going to be your central nervous system versus your peripheral nervous system. Your central nervous system includes your brain and your spinal cord. Okay, so the biggest point here that your course pack tries to make is the clear delineation of where it goes from central nervous system to peripheral nervous system. So here is the spinal cord running through the vertebral column. And off of the spinal cord, you see spinal nerves. As soon as you see a spinal nerve, that is now part of the peripheral nervous system, meaning outside of the central nervous system. Once again, as soon as you see a spinal nerve coming off of the spinal cord, that is the peripheral nervous system. Now, I have this labeled here, but again, we're going to talk about this a little bit later. But the peripheral nervous system, as soon as you come off of the spinal cord, and here we have a spinal nerve, and then your course pack tells you that the anterior ramus of the spinal nerve and posterior ramus of the spinal nerve are considered the peripheral nervous system. And we'll touch on what those mean a little bit more later on. Now, the spinal cord, or the spinal nerves, the peripheral nervous system, is organized as 21 pairs of spinal nerves. A spinal nerve is a nerve that comes off the spinal cord that goes through the intervertebral foramen, which we'll point out in the next slide, to supply different regions of the body. So spinal nerves are named based off of the vertebrae they are associated with. So for the cervical vertebrae, it is named for the vertebrae below the nerve. So C1, the nerve, passes over top of the C1 vertebrae. C3 passes over top of the C3 vertebrae. Now, if you remember back to the video on the vertebral column, there is no C7 vertebrae, right? It goes from C8 vertebrae. It goes from C7 to T1. So the C8 nerve is going out over top of the T1 vertebrae. For all the other spinal nerve segments, so you have T1 all the way to S5. You also have a coccygeal nerve, which I didn't include in this. But from T1 all the way down to the end, those spinal nerves are going to be named off of the vertebrae above them. So for instance, the T1 spinal nerve right here comes out below the T1 vertebrae. The T12 spinal nerve comes out below the T12 vertebrae. Okay, so you have a spinal nerve that is associated with every vertebral level, and they are arranged in 31 total pairs. You also have cranial nerves, and you're going to have 1 through 12, and you'll get very familiar with these in your second unit of the course. So going into a little bit of the spinal cord organization, just a little bit more in depth, what I want to point out here is that intervertebral foramen. This intervertebral foramen is where every spinal nerve is exiting the vertebral canal, which is where your spinal cord runs, to get to the periphery. So the intervertebral foramen. Now going through this term, just so we can get used to it, intervertebral, between the two vertebrae, foramen meaning space. So that is allowing the spinal nerves to exit into the periphery. Now over here on this right image, we're showing a lower portion of the spinal cord. What I want to point out here is that the spinal cord ends at the level of the L2 vertebrae. So that's happening right here. And that is going to be termed the conus medullaris. So the conus medullaris is the end or the termination of the spinal cord. And we'll touch on this again shortly. Further down, you still have spinal nerves that need to exit. So your L4 spinal nerve, for instance, needs to exit underneath the L4 vertebrae. So if the spinal cord ends at L2, how do these nerves get to their target? Well, you're going to have the nerves continue down inferiorly past the end of the spinal cord. So these are the nerves continuing down inferiorly, and then the collection of nerves in this region is called the cauda equina, which means horse's tail. So someone thought this looked like a horse's tail because of all of the fibers, so that's why it's called cauda equina. But the cauda equina is a continuation of the spinal nerves after the end of the spinal cord at the conus medullaris. Going into a little bit more of the anatomy of the spinal cord, we want to look at the spinal cord in a cross-section. So in a cross-section, we are going to have a gray matter in the center of the spinal cord that is in the shape of an H or of a butterfly, however you like to think about this. The wider parts of the butterfly are going to be the anterior horns of the gray matter. So anterior horns in the front, they are typically wider in the spinal cord. In the back, you have posterior horns. They're typically a little bit thinner. So posterior horn, anterior horn. These are gray matter. Gray matter means that these are non-myelinated nerves. In this case, this is going to be nerve bodies. Now, the white matter that surrounds it is going to be myelinated. And these are nerve fiber tracts that are running up and down the spinal cord to get from the periphery to the brain or vice versa. Okay, and now I also want to mention some of the things that you're going to see in this gray matter. In this anterior horn, this is where you're going to have the somatic efferent neurons, which we'll visit again later, but these are your motor neurons. So somatic efferent neurons located in the anterior horn of gray matter. That's where the cell bodies are located. And the posterior horn, this is where you're getting input from sensory neurons. So sensory information is coming in through the posterior. Now, something that we don't see in the cartoon image is over here on the right. This is a histological image of a cross-section of the vertebrae. And specifically, what I'm pointing out here is that you see an intermediolateral cell column. So this is a little protrusion of gray matter that is in between the posterior horn and anterior horn. So in this cartoon image, it would be located right in here. Now this structure is only located in the levels of T1 to L2. So in the levels of T1 to L2, you will have an intermediolateral cell column. The importance of this structure is that this is the location of these cell bodies for your sympathetic efferent nerve fibers. So this is the only place in the entire body where you have the origin for nerve fibers for sympathetics coming from the central nervous system. So again, from T1 to L2. So you only see this intermediolateral cell column from T1 to L2. Now going into some of the coverings of the spinal cord, we termed this coverings of the spinal cord the meninges. And there are 3 layers that we need to know. We're going to start from the most outer layer, which is the dura. So all of these layers, each layer of meninges is termed something mater. So the outer layer is the dura mater. So the dura mater here is your outer and thickest layer. If we go deeper to the dura, we are going to find the arachnoid mater, which is a very thin layer. And the space between the dura mater and the arachnoid mater is the subdural space. If we go below the arachnoid mater, closer to the spinal cord, we are going to hit the pia mater. The pia mater is the deepest layer and in close association with the nerve fibers. In between the arachnoid mater and the pia mater is our subarachnoid space, and our subarachnoid space is filled with cerebrospinal fluid. That cerebrospinal fluid coats the nerves and allows them to float in space, so that way we have a little bit of a shock absorber amongst some of the other functions that it serves. Now, in order to keep the spinal cord in place and not so it's bouncing around so much, the pia mater has some special appendages called the denticulate ligaments. So the denticulate ligaments are a special function of the pia mater, and they go on either side of the spinal cord in a serrated fashion, and at every vertebral level, they're going to punch out and grab a hold of some dura. So they go through the arachnoid and attach to the dura. So these are extensions of the pia mater that help keep the spinal cord in place. Looking over here towards the right, we're looking at the termination of the spinal cord, which we saw earlier. Termination at the level of L2 at the conus medullaris. And what I want to point out here is that this space between the end of the spinal cord down to S2, there is an open space that is the subarachnoid space in this region, and that is specifically going to be called your lumbar cistern. So your lumbar cistern is your subarachnoid space between the end of the spinal cord and S2. So lumbar cistern, subarachnoid pouch between L2 through S2. Other important structures I want to point out in this region, again, once the cauda equina is a continuation of nerve fibers that run through the lumbar cistern. And then lastly, again, to keep your spinal cord in place, you don't want it swinging all around, we want to somehow attach the end of the spinal cord, the conus medullaris, to the sacrum. And in order to do that, we have a continuation of the pia mater that forms a thin band and runs all the way down to the sacrum, and this is going to be called the filum terminale. So the filum terminale is an extension of the pia that runs all the way to the sacrum to keep the spinal cord in place. Now, it's great that we have a spinal cord, but somehow we have to supply it with blood and nutrients. Otherwise, our spinal cord would die, and that would be very bad for us because we wouldn't be able to do the things that we know and love. So, what is the vascular supply of the spinal cord? I want to point out that the spinal cord has one anterior spinal artery. There is a singular anterior spinal artery that runs up and down the spinal cord. This is going to be supplying the majority of the spinal cord. But you do have two supplementary posterior spinal arteries that supply a little bit less, located posteriorly, but there are two of these arteries that run up and down the spinal cord. Now, this artery, this anterior spinal artery that runs up the spinal cord, needs to be supplied from some other branches. So it's reinforced by anterior segmental medullary arteries. You will also have posterior segmental medullary arteries. These medullary arteries are coming in and joining that anterior spinal artery at every vertebral level. So that's what I'm highlighting here. At every vertebral level, you have an artery that's coming in to help supply that anterior spinal artery to make it a little bit more robust. And those are your segmental medullary arteries. Now, it does need to be pointed out that you do have one large medullary artery that supplies most of the lower 2/3 of the spinal cord, and that is your great anterior segmental medullary artery. So once again, this provides a large amount of the blood supply to the lower 2/3 of the spinal cord, and this will become important for a clinical application that we will see later. Now, I want to take a little bit of time with this and talk about spinal nerve anatomy. There's a really good image in your course pack that will tie this concept into the sympathetic nervous system, but I'm going to try to do so with just talking about the somatic nervous system. So we've already talked about your gray matter. You have an anterior horn, a posterior horn, and then you have white matter surrounding. Now, coming off of the anterior horn, you have a ventral or anterior root. Okay, the root is formed up of a bunch of these small fibers, which are called rootlets. The same thing is happening in the back. Coming off the dorsal horn, you have a bunch of small nerve fibers, which are called dorsal rootlets. In the anterior region, once these rootlets come together, it is now called a root. So rootlets form a root. And in the front, this is going to be called the ventral root. In the back, we have dorsal rootlets forming a dorsal root. So if I continue going peripherally, when these two roots come together, the ventral root and the dorsal root, you will form a spinal nerve. So a spinal nerve is a combination of the ventral root and the dorsal root. Now, we mentioned that in the anterior horn, this is where you have efferent fibers, somatic efferent fibers or motor fibers located. Fibers are going to go from the anterior horn, that is the cell body for the neuron, and shoot out through the ventral root. So efferent fibers, motor fibers, go through the ventral root of the spinal cord, but until they join with the dorsal root and is termed a spinal nerve. Sensory information comes through from the periphery, through the spinal nerve, and backwards to the dorsal root, through the dorsal root to the dorsal horn. So sensory information coming from the periphery through the spinal nerve, through the dorsal root to the dorsal horn. That is sensory information are afferent fibers. Afferent fibers going to the central nervous system, efferent fibers going away from the central nervous system. Now, what I want to point out here is we mentioned that sensory nerves are typically pseudo-unipolar neurons. So, the cell bodies are located in a ganglion. And this ganglion is specifically located on the dorsal root. So we give it the name dorsal root ganglion. This is the location of the cell bodies for the sensory nerves that run with the spinal nerves. So that is the dorsal root ganglion. And remember, a ganglion is a collection of cell bodies, of neurons. Okay, so this is and specifically in the peripheral nervous system. And we know we're in the peripheral nervous system because we have exited the spinal cord. Now, if we continue distally, you're going to have a dorsal ramus and a ventral ramus. You could also see this as posterior and anterior, depending on the source that you're looking at. But what's happening here is that you have a bifurcation of the spinal nerve, and one portion is going posteriorly, and one portion is going anteriorly. That is your dorsal ramus going posteriorly, ventral ramus going anteriorly. Now, one thing I want to add in, and again, this is going to be the focus of the majority of the next video, but I want to mention some sympathetics. So, what we're looking at here in this image, let's start with the spinal cord. The anterior horn gives us the ventral rootlets going to the ventral root that is joining with the dorsal rootlets, dorsal root, and together they form the spinal nerve. That spinal nerve here is bifurcating into a posterior ramus and an anterior ramus. Now, what is important to note is that the anterior ramus is communicating with a structure called the sympathetic trunk. The sympathetic trunk, again, we will go way more into this in the next video, but the sympathetic trunk is ganglion for your sympathetic nervous system, and that is part of your autonomic nervous system. And that is, again, something that we will cover much more in depth in the next video. But I want to point out that off of your anterior ramus, you have connections to the sympathetic trunk. A connection to the sympathetic trunk from a spinal nerve is called a rami communicans. So going from the rami and communicating, the anterior ramus and communicating to the sympathetic trunk, rami communicans. If it is myelinated, it is called a white rami communicans. If it is not myelinated, it is called a gray rami communicans. And we'll talk more about this later. But what I want to show in this image to the right is that you have a ganglion for every single level of the spinal nerves. So every single vertebral level has an associated ganglion. And there is a connection between the gray rami and white rami communicans at these levels. All right, now moving into one of the last components of this unit. We're going to talk about plexuses and dermatomes. So what you really need to know about a plexus is plexus is an anastomosis or a joining of a bunch of anterior primary rami of multiple spinal nerves. So within a plexus, different spinal nerve fiber groups can join and regroup to ultimately form terminal nerves supplying the extremities. This is really just introducing the term of a bunch of nerves coming together to form new nerves that we give different names that we will see later when you learn the brachial plexus towards the end of this unit. Lastly, I want to talk about the dermatomes. The dermatomes are areas of skin supplied by the sensory root of a single spinal nerve. So, for instance, your C1 spinal nerve has a sensory component that goes, C1's a bad one. Let's say your C4 spinal nerve has a sensory component that goes to this specific region of skin, and that region of skin is called a dermatome. And what you can see is that the dermatome starts at C2 above and works its way down all the way to S5 back at the bottom of your tail. And what's important to know when you're thinking about dermatomes is you have to understand that when the way that our dermatomes are distributed, you have to think about us as standing on all fours like a dog. So starting at C1 at the top and at our tail, at our sacrum, that's where we're going to get our S5 vertebrae. That's why you might see in the toes you have L5 and S1 as opposed to S5 if we were just going from top to bottom. Now, one other thing I want to point out that this image has reminded me of, C1 does not have a dermatome. And you say, Ryan, how does C1 not have a dermatome? That is because C1 does not have a posterior root ganglion, so it does not have sensory information coming in at the level of C1. All right, last thing before some clinical anatomy, I want to go through some functional components, or really some functional terminology that you're going to be hearing throughout the rest of the course. First, we're going to talk about general somatic afferent. These are going to be GSA fibers. That is going to be how you see them abbreviated from here on out. GSA fibers are somatic sensory fibers, meaning they're from your somatic nervous system. They are sensory and they can sense pain, temperature, and touch. General somatic efferent fibers are going away from the central nervous system. That is what the E is, efferent. They are somatic motor and they're voluntary control. So when I flex my biceps, what my brain is doing is sending a signal through the general somatic efferent pathway, the GSE fiber, to my bicep to contract. When I think about general visceral afferent pathway, visceral meaning our internal organs, so this is going to be part of my autonomic nervous system. Specifically, afferent meaning sensory, so information coming back to the central nervous system. So this is going to supply sensory information to smooth muscles and glands. General visceral efferent fibers, GVE fibers, are going to be motor fibers leaving the central nervous system. That's what the E is, efferent leaving, going away from the central nervous system. And these are going to be autonomic fibers. That is the visceral component. So make sure that you understand these abbreviations, GSA, GSE, GVA, GVE, as they will be coming up again and again throughout the course, and you'll be able to recognize what these mean very quickly. Okay, last little bit of terminology that I want to talk about here is in the periphery, nerves are composed of bundles of nerve fibers that are held together by connective tissue. Right, and this connective tissue is continuous with the meninges in the central nervous system. Now, the first layer that we need to be aware of is the epineurium, epi for above. So epineurium is going to surround an entire nerve. Okay. Now, there are a bunch of different fascicles in here. That is a term that you don't need to know quite yet. But each one of these fascicles, which is a group of nerve fibers, is going to be covered in perineurium. So it goes epineurium is most superficial, perineurium is next, and then each one of these individual nerve fibers is going to be covered with an endoneurium. So epi, peri, endo. Right, and that'll take us to our clinical anatomy correlations. So our first clinical anatomy correlation is going to be a lumbar puncture. A lumbar puncture is a procedure where you're draining CSF fluid from the subarachnoid space to try to analyze for possibly meningitis or other causes. The important thing here is that we're putting a needle into the region where we're at risk of endangering the spinal cord. So how do we minimize this risk? We go to a level that is below the spinal cord, right? We know the spinal cord ends at the level of the L2 vertebrae, so we typically go between the L3 and L4 vertebrae or the L4 and L5 vertebrae. You don't want to go lower than the L5 vertebrae because then you're getting to the sacrum. Now, you might say, well, Ryan, we might hit some of these cauda equina fibers. Good news, if we go in gently, these typically can just be pushed to the sides and we're not going to cause any damage. And again, we're in the subarachnoid space and we're utilizing the lumbar cistern. Okay, so a lumbar puncture happens between L3 and L4 or L4 and L5, and the needle is going into the subarachnoid space and the lumbar cistern to drain cerebrospinal fluid. The next clinical correlation that we're going to discuss is an epidural anesthesia. So an epidural anesthesia occurs by pushing a needle through the muscle and layers of fat and tissue to get to the coverings of the spinal cord, the meninges, but we're going to inject the anesthetic agent over top of the dura mater. So here's our dura mater, which is our most superficial meningeal covering. And here is the anesthetic, agent, right? And that is happening above the layer of the dura. So this puts you at less risk of damaging the spinal cord. Now you have to be careful though, if you go in too deep, what's going to happen? You're going to puncture the spinal cord and you can cause injury. All right, and the final clinical correlation for this unit, we want to talk about the great anterior medullary artery. Now, we mentioned this already. The great anterior medullary artery is one of those medullary arteries that is supplying the anterior spinal artery. So there's the singular anterior spinal artery running up and down the spine, but the great anterior medullary artery is supplying some of the circulation to that vessel. So if you occlude or damage this artery in any kind of surgery, that can cause pretty disastrous events for the inferior 2/3 of the spinal cord. And that'll conclude this video on this course back chapter.